Guys, so now we'll be going over gluconeogenesis. So one thing we do know is it occurs mostly in the liver. Sometimes it happens to the kidney, but sometimes it's a key word, right? So there's some key enzymes involved. Here are the biggest enzymes. So pyruvate carboxylate turns pyruvate. OAA is oxaloacetate, okay? Turns pyruvate to oxaloacetate that then goes to your mitochondria. PEP carboxylase kinase, we just call this PepCK, turns the oxaloacetate that you just made back to PEP in the cytosol. This step is needed because for you to be able to get PEP into the cytosol, you have to make it oxaloacetate because PEP can actually not travel through the cytosol. PEP cannot travel through the mitochondria. So to get your pyruvate through the mitochondria, you would need to first turn it into oxaloacetate. Oxaloacetate can then travel through the mitochondria, where then you have PepCK, which now you can turn the oxaloacetate back into PEP. So fructose 1,6-bixphosphatase is, remember, it's going to be the reverse one from what we have in how did I forget it? Glycolysis, okay? So this is reverse from where we have to glycolysis. So here we're actually going to take the phosphate group on carbon 1 off. This is actually your rate limiting step, okay? So this is the step that sets how quickly gluconeogenesis is occurring. So glucose 6-phosphatase, this is, again, another important thing. You should just know only in the ER of the liver, okay? And again, this is going to be the opposite of what we have in glycolysis. So what we need is gluconeogenesis requires ATP, GTP. It reverses glycolysis using different enzymes at irreversible steps. So what you need to know about gluconeogenesis is it's the formation of glucose, okay? So it's when you take pyruvate, which looks like this, into your glucose, okay? And that happens through gluconeogenesis, okay? So again, reverses glycolysis, uses different enzymes, okay? The reason why I didn't show all the enzymes is outside of these ones that I showed here, the rest of the enzymes are the exact same that you had in glycolysis. So it's not like you're memorizing a whole bunch of new enzymes. These are still the same ones that you use in glycolysis. These 4 are the only 4 that are different. And these 4 are what we call the irreversible steps of gluconeogenesis, okay? Next, we're actually just going to jump right into the pentose phosphate pathway. If there's any more questions about gluconeogenesis, please let me know. So PPP produces NADPH and ribose 5-phosphate. What you need to know is this is actually very important. Ribose 5-phosphate actually makes your nucleotides, okay? And then NADPH is for biosynthesis and antioxidant defense. So some key enzymes in the PPP is glucose 6-phosphate dehydrogenase, which we just call G6PD. You'll see this a lot abbreviated. They don't really. It's annoying because I don't like abbreviations. But anyways, this is your rate limiting step. It is activated by NADP plus and it's inhibited by NADPH. Okay. And function of NADPH is it is the biosynthesis of fatty acids and cholesterol, and it assists in regeneration. So it assists glutathione in regeneration. So NADPH actually acts as an antioxidant, and I know that we've probably all heard that word at some point. So it's actually your NADPH is actually one of those things that acts like an antioxidant. So hopefully that was helpful. And again, any questions, please ask. This is pretty much all Kaplan covers about these stuff. I could get into the nitty gritty, like every step, but I'm assuming that you know a lot about glycolysis. So gluconeogenesis, this is really the only difference that you need to know. And then for PPP, I actually haven't seen a lot of questions about the PPP. So this actually should be just enough to get you through your MCAT. Gluconeogenesis, you may see in more detail. So next, I see you guys in chapter 10, where we talk about acetyl-CoA.